best days of being a PI so far. This is absolutely the best thing about being a PI is mentoring your students and watching them defend their theses. It's pretty amazing experience. So thanks for that, guys. So it is my really great pleasure to introduce to you Mike Cassie, who's going to describe for you his thesis in a minute. And I'll say a few words about Mike. So I distinctly remember during the Penn graduate recruitment back in 2011, I gave this really quick little spiel about sort of what we do in my lab. And I think all I did was sort of show the attention task. And at the end I get this question, this somewhat obscure question about contrast sensitivity, psychophysics or something from the back of the hall. And I thought, oh, this is great. There's a student who actually knows something about vision. And then afterwards, I think during the same weekend, Mike came up to me and said, "Hi, I'm Mike. I really want to rotate in your lab." And I was like, "Okay, I did something right, you know." And as I was saying yesterday before Vanessa's presentation, I really did my best to try to discourage these guys from joining my lab. I told them you're going to have to read lots of papers, you're going to have to learn to write code, and in Mike's case, you're going to be spending all night in the lab. It's going to kill you. And there was nothing for them to do when they rotated because I was just building my lab, and yet they both decided to join the lab, and I'm really glad they did. So Mike is, I think, a rare breed of student who, I think, almost from day one took ownership of his project. You know, there was no lack of excitement and motivation for the work that he did, and that just shone through every day that Mike was in the lab. He really gave it his all every single day. I think that's a pretty fantastic quality in a scientist. So that was a privilege to watch. Mike he has a very healthy skepticism for his data. So there were many days, and this happened many times, where I'd be sitting in my office and Mike would come in and, "I think it's all bad." I think there's nothing here, and I would say, "Okay, let's go and look at the data," and we would go through, and inevitably, no, this is actually really interesting. We just have to look at it this way and to think about it in this way. So a healthy skepticism is good, but don't downplay your data, your results too much. But really, in the end, the work that Mike is going to sort of just give you this beautiful story today, which is. I think it's probably hard for this audience to appreciate what a seminal work this really is for our field. And I also just want to highlight that he's going to breeze through all of these pretty complicated methods, but he really did all of this himself from start to finish. These experiments are brutal, and it's not for the faint of heart. And Mike just dove in and really became completely independent remarkably quickly. So again, pretty amazing qualities for a student. I'm incredibly proud of him. So, with that, take it away, Mike. All right.
Thanks a lot, Karen. That was, as she mentioned, I am very, very proud of this project. I'm really excited to show you the data here. And starting when I walked into her lab in order to do the rotation, she kind of described the idea behind this project to me, and I thought every single bit of it sounded awesome. So I was like, this is what I want to do, and this is what it is. So I'm really excited here to tell you about what we've learned about the role of corticogenicular feedback in visual perception. So I'm going to start off pretty basic, and at its most basic, the role of the brain is to detect sensory information and integrate that into create a relevant motor or behavioral output. All sensory systems across organisms have this some ability to detect stimuli and respond to the external environment. A major limitation across sensory systems is that it's almost impossible for the brain to consciously process all of the incoming sensory information at one time. So, as an example, so what the brain does is it sorts this information into relevant and irrelevant information depending on the organism's situation, which I'm going to refer to as its behavioral context. So, if for an example, right now you're all sitting in your seat, but you're probably not aware of the position of your tongue in your mouth, or how the soles of your feet feel inside your shoes. And now that I've brought those things to consciousness, that information is relevant, so now you can actually think about how those kind of things work. And the visual system is no different, though perhaps it's a little bit more subtle. So if you take, for example, these two situations, this situation on the top, well, first of all, both of these are very highly visual scenes. And what I mean by that is, in order to be successful in both of these endeavors, it requires visual information, basically. So if you're across the street, you basically have to have eyes. You have to be able to pick up that information. And the type of visual information that's relevant in this situation is a little bit different than the type of visual information that's relevant in this situation. As you can imagine, when you're trying to cross a busy street, things like motion and the spatial relationships between objects are more important than when you're trying to paint a painting, where things like form, color, and detail are more important. And the brain needs some sort of a mechanism to separate, the brain separates this information, and it needs some mechanism in order to tell what is relevant for the current situation, and to make sure that the relevant information is maximized in order to be successful at the behavioral context. So in the visual system, in order to give even more background, we have to start talking about how things generally operate. Visual information is processed in these discrete hierarchical steps. That's kind of the general model for the feed-forward flow of visual information. Light energy comes in through the eye, where it's transduced by photoreceptors in the retina, transduced in a neurochemical signal. Retinal ganglion cells here at the back of the retina then project their axons to the thalamus, specifically to the lateral geniculate nucleus, which is the primary visual nucleus of the thalamus. It acts as kind of the first major sensory processing hub for visual information in the central nervous system, and then it relays information onto the primary visual cortex, which you can think of as kind of a very simplified explanation of the primary visual cortex, is that it is a gateway to higher processing areas where this visual information is increasingly integrated eventually with other types of sensory information, ultimately to create a complete perception. In our lab, we focus on the early visual system, which is kind of these initial steps. And I described this as the kind of canonical thinking for how visual information flows through the brain, but this system is not unidirectional. There are actually feedback pathways that exist from higher cortical areas higher in the hierarchy back down to areas lower in the hierarchy. And the pathway that we're interested in is called the corticogeniculate pathway. It's one of these feedback pathways, and these neurons reside here in the visual cortex, and they project back to the lateral geniculate nucleus, the corticogeniculate pathway. And now this pathway is anatomically robust. It was fairly obvious even from the beginning when people started looking at the anatomy of the visual system, but its function has still remained somewhat of a mystery. A lot of experiments have gone into trying to figure out the role of this pathway, both in vision and in other sensory systems. And so far, a definitive functional role for this pathway has yet to be determined. So we study this system. We study it in ferrets. Ferrets are small, they're easy to handle, and they're highly visual mammals, meaning that they use their primary sense is sight. And because of that, they have a well-organized and well-laid-out visual system. It's pretty comparable to that of cats, which are another classic visual model. So ferret LGN, like cat and primate LGN, is a layered structure, and here the LGN relay neurons project to layer 4 of the visual cortex, but these LGN neurons split visual information into discrete streams. So there are multiple types of LGN relay neurons, and the two that I'm going to talk about most in this talk are the Y pathway, and the Y pathway focuses most on motion and spatial location information, whereas the X pathway focuses most on things like form and detail. Experimentally, you can tell these neurons apart based on their responses to when you change different parameters of the visual stimulus. So for example, if we were to show these neurons visual stimuli like these gratings down here in the corner, if we were to show a grating that then increases its contrast in certain increments, you can plot what's called a contrast response function, and that's what I have down here. And one of the ways we can tell these neurons apart is that Y cells are sensitive to lower contrasts, and they end up having this nonlinear contrast response function, whereas X cells are sensitive or have this very linear contrast response function. So by plotting these measures, we can tell the neurons apart right after we have recorded them. As I said, the feed-forward information flow from the LGN separates information into these discrete X and Y pathways. That information, LGN neurons then synapse onto layer 4, which is the primary thalamic input layer of the visual cortex. That information kind of bounces around in the cortex before it leaves either via layer 2, 3, or via layer 5. I also, as I mentioned before, there are these cells here in layer 6 that receive the majority of their input from layer 4, and they project back to the lateral geniculate nucleus. So they project from the cortex to the geniculate, forming the corticogeniculate cell path. There are a few interesting things about the corticogeniculate cell pathway. The first is that the corticogeniculate synapses onto LGN relay neurons greatly outnumber those of the retinogeniculate inputs. This is almost 10 to 1 across most species. Despite that, LGN neurons inherit this center surround receptive field shape from their retinal ganglion cell inputs, rather than these more oblong orientation-tuned receptive fields that you would see in the cortex. One of the reasons for this are that if you can look at the anatomy and you can see that corticogeniculate synapses, as shown by this schematic here, tend to be small and they tend to be out on the distal dendrites, whereas retinal ganglion cell synapses tend to be large, robust, and they synapse a bit more proximally, closer to the cell body. And these synapses are capable of generating the kind of driving input that influences receptive field shape. So it's pretty clear from the anatomy and the anatomy and physiology of these synapses that corticogeniculate modulation of LGN relay neurons is pretty subtle. So even though it's an anatomically robust pathway, it's kind of physiologically weak. And I think that's one of the reasons why the actual defined role of this pathway in the cortical circuit has remained elusive for so long, is that its role is subtle. So our experiment design is actually pretty simple. We want to record the visual responses to LGN relay neurons, see how they respond to visual stimuli, and then directly stimulate the corticogeniculate pathway and record the responses to those LGN neurons to the same stimuli while you have corticogeniculate input, just to see how those responses change. The way we go about doing this is we inject a genetically modified rabies virus. So this SAD delta G virus is missing an important membrane-bound protein that allows it to cross synapses and spread the infection like wild-type rabies does. Instead, this virus expresses the genes for a light-sensitive ion channel. This is called channel rhodopsin. And channel rhodopsin is an ion channel that will open in response to 480 nanometer light. And when it opens, it causes the cell to depolarize and then to fire. So what that means is neurons that express channel rhodopsin can be driven with light. This virus also expresses mCherry, which is a red fluorescent protein, and that allows us to visualize the infected neurons because they will be glowing red under a fluorescent microscope. So we inject this virus into the LGN. By nature of rabies is taken up by the axon terminals. It travels retrogradely up the axon of cells where it infects cell bodies. It's important to note here that LGN neurons do not, the LGN relay neurons do not have vocal axon terminals within the LGN. So they will not pick up the virus and they are not infected with channel rhodopsin or mCherry. It's also important to note here that by definition, any neuron we see any neuron with axon terminals in the LGN, and by definition, any neuron that's then infected in the cortex has to be a corticogeniculate cell, because no other cortical neurons project to the LGN, and no other cortical neurons can pick up the virus. And then because rabies can't move transsynaptically, it only infects there's only the initial infection, it can't spread. So when I got these injections right, it actually worked pretty well. This is a coronal section of ferret LGN. The LGN is outlined by this dashed line. You can see the injection site, which is indicated by this black arrow. This little arrowhead shows a lesion made by one of our recording electrodes when we were actually recording. And here on the right, this is also a coronal section, but this time this is ferret visual cortex. And you can see here that the infected cell bodies of these neurons are entirely within layer 6, which is consistent with the anatomy of the corticogeniculate pathway. And these neurons look like typical layer 6 pyramidal cells, where they have kind of a pyramidal shape, and their apical dendrites come up with a very dense dendritic ramification in layer 4, some of them extending all the way up into layer 2. This is a more zoomed out coronal section showing that sometimes we get these very robust infections where you can see these very densely labeled patches. And if we get lucky, we're actually going to be recording from one of these densely labeled patches, as indicated here by an electrode lesion on our slice. We also know that you do not get expression of this virus in the visual cortex when you inject into the somatosensory cortex, or the hippocampus, or the ventricles, or the other parts of the thalamus. So we had a fair number of control experiments. We injected the virus and we did the recording in the experiment, but it turned out there was actually no rabies in the actual cortex. But we could use that data then to compare with these experiments. So following the injection, we do our recording, and the experiment is the animal. This takes place 7 to 12 days after the initial injection. That gives time for the virus to metabolize and for the infection to become at its appropriate strength. We anesthetize our ferrets, and we display visual stimuli like this drifting grating on a computer monitor. And we record simultaneously from the LGN and from the cortex. We try to record from what are called retinotopically aligned neurons, and that means that the neurons in V1 have overlapping receptive fields with the neurons we're recording from in the LGN. We try to make sure that those receptive fields are overlapping. This is for two reasons. One is because it makes it easier to drive both populations of neurons with one visual stimulus, and the other reason is that those neurons are more likely to be functionally connected based on the functional organization of the visual system. So we record these visual responses to these kind of stimuli, and then we do the exact same thing, record from the same neurons to the same stimulus, only this time we flash LED on the cortex using a fiber optic cable, and that then drives the light-sensitive infected corticogeniculate neurons. And we can then compare those responses, these LGN responses, with the non-LED or less corticogeniculate condition. This is a panel showing the activation of our light-sensitive neurons. So this figure is the same that I showed you earlier. This is a coronal section of ferret V1. The cortical layers are indicated here by these lines. This schematic here is a illustration of our recording probe in V1. So this probe has 24 contacts, and these contacts are all arranged in a linear fashion. Each contact is capable of picking up extracellular spikes. And then you can look at when we show LED when we flash LED on the cortex, which is indicated by these blue arrows here, we can plot the light responses for these neurons. So these histograms, the 7 histograms here represent the responses of neurons on the deepest 7 channels. So in this case, these are the channels that would be in layer 6. And you can see here that the orange histograms have an elevated response following these pulses of light. You can also see that there are some channels here in layer 6 that do not have an elevated response to light. And these neurons presumably are not infected. They don't have expressed channel rhodopsin, so they're completely insensitive to the light. And this could be either because they are not corticogeniculate neurons, they project elsewhere they project elsewhere subcortically, or it's possible that they are corticogeniculate neurons, but they just didn't project to the injection site in the LGN. But regardless, we have light-responsive neurons, which are presumably corticogeniculate, and we have light non-responsive neurons, which are presumably not corticogeniculate. And this is quantified here on the right, where this is a LED tuning curve. You can see that our light responsive neurons have a meaningful effect, but our LGN neurons and our V1 neighboring neurons do not have any meaningful effect. Again, quantified in this histogram here. So after we demonstrated that we can actually drive our corticogeniculate neurons with light, one of the first major things we wanted to do was we wanted to see how corticogeniculate feedback changes the receptive field properties of the LGN neurons. So a visual neuron's receptive field is what primarily gives the neurons its response properties. And so changing properties of the receptive field is a powerful way of changing the response properties of the visual neuron. The way we do that, we show a visual stimulus called a M sequence. This is a pseudo-random white noise stimulus. This thing will flash on the screen in the receptive field for a long time. For our recordings, these were almost about 20 minutes. And we were recording spikes from our cell the entire time. Later, offline, we can then take our spike train and we use a process called reverse correlation to create what's called the spike-triggered average. And the basic explanation for this process is every time in our spike train that there's a spike, we take the frame that immediately preceded that spike and we average those frames together, pixel by pixel, to create a kind of spatial luminance map that would be the average stimulus that triggered a spike. And if you do this, ideally the average stimulus that triggered a spike will look something like that neuron's receptive field. So this gives us an idea of what the receptive field looks like in space, and we can also see how the neuron's responses change over time by taking frames further and further away from the spike and averaging those together. As you can see, frames that are very far away from the spike may have no response, but frames that are a little bit closer might start to have some kind of a response. So you end up with something kind of like this. These are spike-triggered averages that show the spatiotemporal receptive fields for our LGN neurons with and without corticogeniculate feedback. So, to walk you through this, for both of these, this is one example on neuron, and this is one example off neuron. And to put it simply, on neurons are neurons that will respond most to increments of light in its receptive field, whereas an off neuron is a neuron that would respond to decrements of light in its receptive field. In both panels here, the top row represents the frames of the spike-triggered average, starting with farthest away from the spike on the left. This was around 60 milliseconds preceding the spike on the left, and immediately preceding the spike on the right, about 20 milliseconds preceding the spike. The starred panels represent the peak frame, which we defined as the frame that has the peak pixel brightness of this measure. So what that tells us, and the interesting thing here that should be kind of immediately obvious, is that without LED, this peak frame occurs well before the actual spike, almost 60 to 80 milliseconds before the actual spike. This tells us that the neuron is very sluggish in responding to this visual stimulus in its receptive field. It's taking almost 60 to 80 milliseconds to respond. Whereas if you compare that with the LED condition, so this is with LED driving corticogeniculate feedback, that latency then drops significantly. So now the neuron is taking around 20 milliseconds to respond to that, to the stimulus in its receptive field. The same thing is true for this example neuron down here. You can see that this off neuron, same thing, takes around 80 milliseconds to respond without the LED, and then with corticogeniculate feedback, it takes between 20 and 40 milliseconds to respond. These figures here on the right are graphical representations of the same thing. So if you look here, the arrows represent the starred frame, or the timing of the starred frame, so the peak pixel brightness. And you can see that there is a very clear shift in the response latency with the LED. So, sorry, the black curve represents with or without LEDs, the blue curve represents with LED. You can see here for this neuron, the peak pixel brightness is somewhere, it takes a much longer time to respond than once we shine LED on the cortex, where that response drops significantly. Same is true for this off neuron down here, however the latency shift isn't quite as robust. There's one more interesting thing that can be gleaned from this data, and that is about, we know that driving corticogeniculate feedback with the LED decreases the response latency, the neurons seem to be firing faster. We can also tell something about the precision. If you see here, in these frames, there's kind of a smearing of the receptive field across these frames in the top row. You can kind of see these little ghosts of the receptive field as we average closer and closer to the spike. This is likely because that neuron is firing, the spikes are occurring kind of sluggishly with a high amount of variability. However, when you come down and look at these frames with the LED, that smearing pretty much disappears, and the neuron responds pretty fast and consistently without the LED, or with the LED versus without. And that's shown here in these curves based on the width of these response curves. So you can see that in both cases, the width of the with LED curve is significantly more narrow than the width of the without LED curve. So we were pretty excited when we saw this. This was something we realized we were actually onto something important here. This is population quantifications of those same things. So those were all example neurons. And here, this is the distribution of our peak response times. You can see, importantly, that none of the neurons in our population had a positive shift. So every neuron had a reduction in latency. Every neuron we recorded got faster. That's quantified here with the peak impulse response. You can see that with LED, there's a significant reduction. And again, there's a significant increase in the precision, which is shown here by this reduction in the width of those curves. So this is a measure of the full width at half height. As you saw, this is these dashed lines. So as I said, this was pretty exciting. When we found this, we thought we were actually onto something. So we decided to test it a little bit further, and this time we went to grading data. So in these figures, A and B are the responses for one Y neuron to 20 presentations of a phase reversing grading. So each row here is a trial. Each dot represents a spike across time. And then these histograms down at the bottom are the summations of all the spikes at any given time bit across time. And this is without LED on the top and with LED on the bottom. And what you can see here is that with the LED, this neuron responds, one, the width of these columns is visually narrower. So these cells, the variability of the spiking time is decreasing. And also you can kind of see that the initial timing of that first spike is also more consistent, and it's bumped a little bit towards the left. This is a little bit more obvious when you look at the PSTHs. So if you look at this first histogram, there's not really much of a peak, and the whole thing is wider than down here, where the peak is shifted a little bit to the left, and the entire thing is a little bit more narrow. So we can quantify this for our population of X and Y neurons. So we quantify the response latency, which is the timing from stimulus onset to the occurrence of the first spike in every trial. And for both X and Y neurons, you see a significant reduction in that response time with LED. You can also quantify, we also quantified the precision. And we quantified that by calculating the standard deviation in the timing of that first spike. So we calculated the standard deviation in the timing of when that first spike occurred. And you can see again that there is a significant reduction in that variability, which indicates an increase in the precision of the neurons firing. So what we see here is with LED, so with the LED driving corticogenicula feedback, we have LGN neurons that are responding faster to visual stimuli in the receptive field and more precisely. One of the things we wanted to test then was, because this is a circuit, if you remember that schematic, LGN neurons feed into the cortex, and then those corticogeniculate neurons feed back onto the LGN. So now we wanted to record what happens to certain V1 neurons when we're driving with LED. And so these are the responses to V1 neurons in layer 4, which would be the thalamic input layer. And what you see here is that there is a reduction in response latency that's not significant. However, there is a significant reduction in that first spike standard deviation, indicating that there's a statistically significant increase in the precision of these V1 responses. There's a fair amount of data in the literature that suggests that this corticogeniculate feedback pathway changes the responses of LGN neurons to different tuning parameters, particularly what they would call a gain change, where the neurons with overall response is elevated, but it doesn't change the tuning parameters. So we decided to look at tuning of LGN neurons as well. And so these are population tuning curves for Y neurons on the top and X neurons on the bottom. These are contrast response functions. So for this plot, we varied the contrast of our drifting gratings. They increased their contrast incrementally. In these figures here, we increased the spatial frequency of those gratings, and you can think about that as the width of the bars that are in the grating. So the bars started out very wide and got smaller and smaller and smaller. And then lastly, we did the temporal frequency, which is just the speed at which these gratings drift. And what's important to see here is that with LED or without LED, these tuning curves look the exact same. There's no major effect in any direction. We don't see a change in tuning preferences. None of the response curves change with the LED, and there's no significant excitation or elevation of the response with light, except for the Y neurons with temporal frequency. And at first, this was very interesting to us, so we took a closer look at it. This is a quantification across our population. This is the max firing rate or max spiking rate for LGN, X, and Y neurons, which would just be the peak response of these curves. You can see that for Y neurons, it's significantly elevated with the LED, but for X neurons, it's not. And then we also looked at the magnitude, which is just the integral beneath these two curves. And you can see again that there's a significant increase in the integral between these curves for Y neurons, but not for X neurons. One of the concerns we had came across because of the design of our experiment. We wanted to avoid disrupting the visual responses of these LGN neurons with our LED. So we flashed the LED at the frequency, the temporal frequency with which the grating was moving. So we aligned the flash with the temporal frequency. For these plots, that flash frequency is held constant. But for the temporal frequency curves, as we increase the rate of the grating, we also increase the rate of the LED. And as you might imagine, if you flash the LED more, you might be driving the neuron more artificially by light rather than any kind of circuit-based visual response. And indeed, we do think that some of this effect may be because of the summation of those LED responses. But we also think that this is pretty interesting, and this might highlight some of the differences between LGN, X, and Y neurons, because Y neurons seem to be able to respond to that increase in light, the increase in frequency, whereas the X neurons don't seem to be able to respond quite as well. So this might indicate some kind of changes or differences in the cellular machinery, perhaps receptor expression, that allow the Y neurons to follow higher frequencies than the X neurons. And then so down here, these plots are just to demonstrate that our tuning preferences do not change. So we show the tuning metric with the LED versus the tuning metric without the LED for X neurons in pink and Y neurons in green, and there is no change for contrast, spatial frequency, or temporal frequency. As I said, we found so far that corticogeniculate feedback seems to be increasing or decreasing the response latency and increasing the precision of these LGN neurons without significantly changing the tuning properties. And the anatomy of the circuit allows us to speculate why we think that may be. So in addition to corticogeniculate neurons targeting the LGN and LGN relay neurons, they also target the inhibitory envelope called the reticular nucleus. And the reticular nucleus is, as I said, is this big envelope of inhibitory neurons that provides the majority of the inhibition to most sensory thalamic nuclei. And what could be happening here, what we suspect might be happening, are that corticogeniculate cells are directly exciting the LGN relay neurons, and that direct excitation is just enough, it's subtle, but it's just enough to bump up the excitability. Maybe it's increasing the membrane polarization, just enough so that when the visual information, the visual stimulus comes online, those cells are primed and ready to respond right away. And then, simultaneously, these neurons are also exciting the inhibitory neurons of the reticular nucleus, which then after some short time delay, come back and inhibit the LGN neurons, which would kind of cut off those sluggish spikes. So the initial feedback is exciting, which would cause an increase in response or a decrease in response latency, but then we have a slightly slower disynaptic inhibition that comes online to clean up the spiking patterns and make these things a little bit more precise. Since our experimental setup isn't really set up to test this directly, we created a fairly simple linear model in order to try to get some mathematical evidence in support of our little hypothesis here. So this model ultimately shows that small increases in corticogenicular activity can lead to the shifts in LGN visual responses that we see. And to describe this here, we did this in 2 steps. First, the model is basically just that the sum, or the LGN response is the weighted sum of the retinal ganglion cell, corticogeniculate cell, and inhibitory inputs with some time delay. So we used our LGN data, first without LED, and we used retinal ganglion cell data from the literature, our own corticogeniculate data, and inhibitory data from the literature, and we left the weights free, and then just tried to solve this model to minimize the variability between the model's output and our real output. Then, once we determined the weights for the retinal ganglion, what the appropriate weight for the retinal ganglion cell, we then fixed that weight and ran the model again, paying close attention to the weight of the corticogeniculate feedback. And what we end up finding is that small increases in the weighting of that corticogeniculate input are enough to lead to these slight shifts in the timing of the LGN responses that you see. So this black curve would be the LGN response without LED, and then this blue curve is the LGN response with LED. And that's kind of shown here in this little schematic where without the LED, or rather I should say with the LED, the weighting of that corticogeniculate input is increased, which is shown here by the thicker axon. The most interesting thing about this is that the model predicts a very modest increase in the weight to lead to a shift like this, around 6%. So about a 6% increase in the corticogeniculate input is enough to cause this timing shift. And that is very consistent with our actual data. So when we drive our corticogeniculate neurons with light, we see between 6 to 11% increase in the corticogeniculate firing rate. We thought this was pretty cool, and it's kind of some mathematical evidence to support that our disynaptic inhibition hypothesis is plausible. So, as I've shown you in this first part, we think that corticogeniculate feedback is increasing or decreasing the response latency and increasing the precision of these LGN neurons firing. And we think that it might be happening through this disynaptic inhibitory mechanism. The next question then is, if you recall, I said that LGN neurons bring their information in these feed-forward streams. So there's the Y pathway, which is separate from the X pathway. So then the next question then becomes, is corticogeniculate feedback the same? Does corticogeniculate feedback maintain the separation of these information streams, or is it more homogeneous across the entire LGN? And that leads to 2 kind of functional hypotheses, alternative hypotheses for that are testable for how corticogeniculate feedback is made. The first is that corticogeniculate feedback is functionally homogeneous. In this hypothesis, you might expect that there's maybe one cell type, morphological cell type of corticogeniculate neuron that feeds back indiscriminately onto both LGN X and Y neurons. Since we know that corticogeniculate feedback doesn't really change the tuning properties of LGN neurons, you would then expect that if the one neuron is feeding back onto both X and Y, its tuning properties would have to be somewhere in between so as not to change either one too much. And that's illustrated here by these TF and size tuning schematics. The other hypothesis is that corticogeniculate feedback is stream specific. So in this hypothesis, you might expect that there are multiple corticogeniculate cell types which feed back specifically onto LGN-X neurons and LGN-Y neurons. And in order for these neurons not to change the tuning, you might expect that their tuning actually mirrors that of the LGN-X and Y pathway. So you would have corticogeniculate X-like and Y-like cells where their tuning properties are similar to those of their LGN inputs. And there's a spoiler alert. Most of the data in the literature already supports this stream specificity hypothesis. For instance, we already know that there are a fair number of separate morphological cell types of corticogeniculate neurons across in other species. So we set to confirm and clarify those different cell types within ferret visual cortex. And we started off by reconstructing the morphologies of our infected neurons. All of this reconstruction that I'm going to show you here was done by Elise, our lab manager. So she digitally reconstructed these neurons by using software to trace all the different processes, and that gave us 4 distinct morphological types that we see in our population. These morphological types they do mirror the morphological types seen in other species, particularly in primates. So what we have here, we have short pyramidal neurons, we have stellate neurons, we have large tufted neurons, and then we have tall pyramidal neurons. And in order to confirm our visual observations of these separate morphologies, we performed some analyses of these. This dendrogram represents the output of a cluster analysis where we took 6 separate parameters of those neurons. Let me check my notes here. We used the tallest point on the neuron. We used the total number of dendritic endings on these neurons. We used the total percent of the basal dendrites in the white matter. So basal dendrites are these little ones down here. We used the total percent of the basal dendrite in layer 5. And we use the total percent of the apical dendrite up in layer 2-3, and the total percent of the apical dendrite in layer 5. And what this tells us is that when we put these 6 metrics into a cluster analysis, it breaks them up into 3 distinct clusters. It's likely we have tall, we have short, and we have stellate. It's likely that these large tufted neurons, we just didn't have enough. The sample size wasn't large enough for them to separate. I think it's likely that this column here that is classified as tall neurons are actually these large tufted neurons. It's pretty clear that the morphologies of these cells are a little bit different. Also, we did an independent analysis where we looked at we saw that you could separate these 3 clusters. They still appear when you do a principal components analysis. So plotted against the first 3 principal components, these 3 clusters still do appear. This is all evidence that, yes, in fact, these are morphologically separate subtypes rather than something else going on where these are the same subtype and they just kind of look different. These are statistical comparisons for those different, some of the initial metrics that we used in the cluster analysis and for a couple of extras. So for instance, we have, in A, we have cell body position as plotted as the percent in the depth of layer 6. And there are a few important differences that I want to point out in this plot. The first is that the stellate neurons, well, the short and tall pyramidal neurons, there's not much difference in the distribution of their cell bodies throughout layer 6. However, these stellate neurons are very, very deep in layer 6. In fact, the majority of them may actually even be down into the white matter. And that's shown here, that's shown here as the percent of the basal dendrite in white matter. These stellate neurons have a much higher percentage of their basal dendrites in the white matter, and that's presumably because their cell bodies are down in the white matter as well. We also have some differences that I want to point out in the apical dendrites of the pyramidal neurons. So the stellate neurons do not have an apical dendrite. They were excluded from this analysis. The first is that tall neurons have a much greater percentage of their apical dendrite in layer 2-3 than short neurons. Short neurons don't extend up into layer 2-3. The other difference here is kind of interesting is that the short pyramidal neurons have a much greater percentage of their apical dendrite in layer 5 compared to the tall neurons. And all of this is statistical evidence saying that indeed the visually observed morphological subtypes are indeed separate clusters that we can analyze. We think that there are probably more than 3 subtypes. These are some rare and interesting cell types that were observed as we were doing these reconstructions. This one here on the left is rather bizarre. It seems to have 2 apical dendrites extending out in sort of a U shape, and then it has these processes that may be basal dendrites, or they may be branches of the apical dendrite. As far as I know, this cell type has never been seen before in the visual cortex across any species. So what we'd like to do is find more of these and reconstruct them and try to figure out if they're indeed a new cell type or if they are just a weird variation of something that already exists. This neuron here is another example of a weird cell type. You can see that this neuron, it looks like the apical dendrite extends off to the side and then takes almost a 90-degree turn to head towards the surface of the brain, which is up here towards the higher layers. This is a very classic example of a tilted neuron, which have been described in primates. So we didn't see enough of these to reconstruct them to add to our analysis, but it does seem pretty clear that there are tilted neurons in ferret cortex as well as we see it in primate cortex. So as I said, for the stream specificity hypothesis, you predict multiple morphological subtypes. Indeed, we have multiple morphological subtypes. So then the next question is, can you separate, do corticogeniculate neurons have tuning properties that mimic those of the X and Y inputs? And the answer is yes, in fact they do. These are two example neurons. So these were corticogeniculate neurons we identified based on their light response. So we know that they were infected and had a light response. And you can see that very clearly the Y-like and X-like neuron, or the Y-like neuron has a nonlinear contrast response function, much like LGN-Y neurons. And the X-like corticogeniculate neuron has a linear contrast response function, much like X-LGN neurons. Same thing is true down here for this temporal frequency plot. The X-like neuron prefers lower temporal frequencies, just like LGN-X neurons, and the Y-like corticogeniculate cell prefers higher temporal frequencies, just like Y cell, or just like LGN-Y cells. So the real meat of this experiment is whether or not these neurons are functionally connected. And we went about determining that by correlating the spike trains of simultaneously recorded LGN and V1 neurons. So if you recall, I mentioned that we tried to make sure to record in retinotopically aligned areas. So we tried to make sure we were recording from retinotopically aligned neurons. This figure here shows, the dark circle is an aggregate V1 receptive field, and the smaller circles are the location of the LGN receptive fields, the centers of the LGN receptive fields that we recorded from. So you can see we aimed for a high amount of overlap, and we were, for the most part, very successful. Then, offline, what you do is you take the spike trains of each of these neurons, and you cross-correlate them and look for peaks in the corellogram. We specifically looked for peaks in the corellogram that were sharp, that were shifted significantly from 0, and that where more than 2% of their spikes had a correlation greater than 2 standard deviations above the mean, which is indicated by this dashed line. We identified a bunch of functionally connected pairs, 58 total pairs, that had a peak in the correlation in the cross-corellogram that fit our criteria. And then we went back and calculated and looked at the tuning physiology of these neurons, and we determined that the vast majority of them can, in fact, be classified as X and Ys, and that the majority of them are between neurons of similar physiology. So these two examples are these are neurons of Y to Y, and this is an example of an X to X pair. You can see here that this pair here is another probably significantly connected neuron. We didn't have enough tuning data to actually classify these neurons as X-like or Y-like. This was a pretty big deal for us. We can show here that almost 90% of the population of these connected pairs are connected between neurons of similar physiology. A small percentage, 11% of them were between neurons of opposite physiology. But then if you look here at the distribution of the peak latency, so this is the distribution of the shift from 0 of this peak, you'll see that the majority of those opposite pairings were actually positively shifted, which suggests to us that these are in the LGN to cortex direction rather than the cortex to LGN direction. So these are probably feed-forward connections rather than feedback connections. There are a couple of other very interesting things here. One is that if you look at the distribution of our Y to Y pairs in terms of their peak latency, the majority of those neurons have a peak latency between or less than 10 milliseconds. And this is very interesting, one, because these are Y to Y, so Y neurons have low contrast sensitivity and they prefer higher temporal frequencies. And we know from previous data in the primate that neurons, that corticogeniculate neurons with the fastest conduction latencies, so the fastest conduction velocities from cortex to V1, or sorry, from cortex to LGN, are also sensitive to low contrasts and high temporal frequencies, which is consistent with our finding here that the Y pathway has peak latency shifts here of less than 10 milliseconds. That one might align, and we might be able to infer here then that our Y population is homologous to those presumably magnocellular, which is the Y pathway homolog in primates, the magnocellular projecting fast conducting corticogeniculate axons. Also very interestingly here is if you look at the X population, the majority of these X neurons have latencies somewhere between 10 and 15 milliseconds. And this is very similar, again, to data that we see in primates, where the presumptive parvocellular projecting neurons, the ones that have kind of a middle-of-the-road response latency, or conduction velocity, somewhere between 7 and 15 milliseconds, are not sensitive to contrast, but they're sensitive to low temporal frequency, and they are more sensitive to color signals, all of which are hallmarks of the parvocellular pathway, which is the primate homolog to the X pathway. So what we think here is that Y neurons, this evidence seems to confirm the inferences or the assumptions that the fast conducting axons seen in primates are actually the magnocellular projecting neurons, and that they are homologous to these Y projecting neurons in ferrets, and that these X neurons may have similar conduction velocities to the presumptive parvocellular projecting neurons in primates. This is pretty exciting, we thought. Lastly here, I wanted to show you these are example tuning curves for those cross-parallelogram pairs that I showed you in the beginning. So these, the important thing to see here is that the dashed line is the LGN tuning curve, and the dotted line is the corticogeniculate tuning curve. And you should see that there are no statistical differences between those tuning curves. So this is, again, more evidence that our pairs can be classified as X-like or Y-like, and that the pairs exist between neurons of similar physiology. So in conclusion, to sum up this whole process, what we've determined here, we think, well actually I should say, this is what we think is going on. We know we have multiple cell types of corticogeniculate neurons. We know that we learned that corticogeniculate feedback seems to decrease the response latency, so it makes these neurons fire faster, and it increases the precision of those responses. And we think that based on our this process happens through a potentially through a disynaptic inhibitory mechanism where direct excitation of LGN relay neurons bumps up the excitability, causing those neurons to respond a little bit faster. And then the disynaptic inhibition comes on to clean up the sluggish spikes to increase those neurons' precision. And finally, our functional connectivity data suggests that this process occurs in a stream-specific manner, so that there are feedback neurons that do this specifically for the X pathway, and feedback neurons that do this specifically for the Y pathway. And so the implications are, if you go back to this visual scene, if you remember, this visual scene requires a much greater, or it requires the motion and spatial information more than form and detail. Since we know that the Y pathway, or the magnocellular pathway in primates, since humans are primates, carries that kind of information, what we might have here is a mechanism by which the brain, or the visual cortex, can selectively increase the salience of one of those information streams over the other. And that might help you that might help the speed of the processing of the relevant visual information to better complete your task, which is presumably to make it across the street without becoming a hood owner. So with that, I have some thanks that I want to give. We had a bunch of people give us money for this, including the NIH and the Whitehall Foundation. I'd like to thank specifically the Callaway Lab. They gave us the initial batches of virus for us to use for our initial experiments. And we actually got it to work, so then we continued to buy it from them for our subsequent experiments. I'd like to thank Doctors Karen Booty and Kirk Maurer. They're the veterinarians here at the medical center, and they were very, very helpful caring for our animals and preparing for surgery and assisting us during the surgeries and things like that. We give very special thanks to Elise Bragg. She's our lab manager. She did a lot of those, or all of those neuron reconstructions that I showed you in the second half. And she's basically the backbone of the Briggs Lab. When she goes away, things seem to fall apart. When she went on maternity leave a few years ago, I realized that I basically have no idea how my lab works. The operating room would be magically prepared, and all I'd have to do is get to work and then go downstairs and we'd be all ready to go do surgery. And then when she was gone, all of a sudden that didn't happen anymore. I had to learn the hard way. I'd like to thank the Briggs Lab members, both past and present, Jackie and Vanessa, for being very supportive. And I'd like to particularly thank Aditi, Jaime, and Steve. They were undergrads and rotation students who came through and they helped me out by doing a lot of the grunt work that I didn't want to do. And they stayed up with me late at night. Farron kind of alluded to this, but these recording sessions last for 24 hours straight. So, starting with the initial surgery of placing the electrodes, going all the way through, these experiments go on a long time, and you can't really leave. There aren't very many breaks that you get, so it is a fairly exhausting task, and they stayed up late with me many of these nights and made it a lot more fun. I'd like to thank my thesis committee for giving me helpful comments and insightful comments along the way. And I'd like to specifically thank the PEM program, particularly the students. And you guys have been some of the best friends and colleagues that I think anybody could ask for. And the sense of community here is so strong. Everybody is very supportive of each other. There's almost no competition. And I picked up on that when I came here, I think. And I think that's a really big reason why I ultimately chose Dartmouth to do my graduate work. The other reason is that I didn't get accepted anywhere else. And lastly, I'd like to thank Farron. Farron, you are a fantastic mentor. She is very knowledgeable, very skilled. She taught me all of this stuff. I came here as a psych major with one neurobiology class in my resume, barely enough to even be qualified for the PEM program to apply to it. And she turned me into the ferret neurosurgeon and neuroscientist that I am now. And she's also very, very, very patient. I think I can remember being yelled at one time, and I'm pretty sure I deserved it way more than that. And with that, I'm happy to take your questions. Yes?
So, Michael, you'll get an easy one. Why do you all use ferrets instead of cats?
Mostly that ferrets are a lot easier to handle. They're cheaper to have, they're cheaper to keep. Ferrets have a bit less of an emotional attachment than cats do for a lot of people, so it's a little bit easier to sell this work. But otherwise, their visual systems are very similar, so we don't really gain much by using cats versus ferrets from this experience.
So, you mentioned you were a psych major. How would a defect in this sort of regulatory pathway that changes the latency of perception, which you described very well, impact a person who was in a situation like this? What would happen if something went wrong?
Since this modulation is very subtle, I think unless the circumstances were perfect, a defect in this pathway probably would not influence their life on the whole. But I could imagine a situation where somebody has to react very quickly to a situation, maybe while they're crossing the street, and they need the ability to be able to process that there's something moving towards them very fast. And the difference between a half a second means the difference between getting hit by a ball or a car or a baseball pitcher. On the other end of things, I can imagine that maybe people who are who do this a little bit better, perhaps that might contribute a little bit to things like to making fine art. That's a bit of a stretch considering that this all happens within the first 100 or 150 milliseconds of visual processing. It's possible.
Question 3.
Yes.
No question, that's it.
Yeah.
Follow up on his question is this: do you think this is a tunable parameter, an artificially tunable parameter, so that if I wanted to say have something where visual acuity at the millisecond level is important, say hitting a fastball, is that something that, are we suddenly going to start having to do testing for your kind of
For
feedback
neural
pathway drugs?
enhancements? Not immediately. But yeah, I think it would be plausible that we could develop some kind of a stimulator that could directly stimulate the feedback pathway in order to maybe increase your perception at the millisecond level. It would be very, very, very hard though, because one of the reasons is that this all takes place at a retinotopically aligned level. So I'm not selectively, this is something that I didn't really get into much, but we're not just selectively increasing those information streams globally across the visual field. This is happening at the level of individual receptive fields, or rather overlapping receptive fields in the hierarchy. So in order to have some kind of stimulation like that, your system would have to be able to figure out what receptive field and which particular neurons feed into that receptive field in order to be able to stimulate it. You might be able to increase it on a global level by having some kind of an implant that just dramatically increases the tone of all of the neurons in that area. But that was one of the problems with the initial research into this pathway to begin with. Was there are a lot of other cell types in layer 6, and those cell types do other things. They go other places. So kind of a global enhancement of that activity might cause more harm than good. I hope that answers your question. Yeah. Yeah.
So these two experimental design, these were all experimental naive animals, right? So you're testing kind of basal speed of their visual acuity and response. So
Yes.
do you have any idea of how this changed the plasticity over time with training protocols?
That's a good question. My guess is that you do see typical synaptic plasticity measures, so there's probably some kind of potentiation that occurs in this pathway in receptive fields that are stimulated more often. I would guess, I don't know this for sure, but I would guess that specifically in the fovea, which is the region of our eye that has the highest acuity, so that's where we tend to focus our vision. Those neurons are probably have greater synaptic efficacy than periphery that aren't used quite as often. In terms of the mechanism for how it happens or what changes occur with training, I would guess that they are, if they do happen, that they are probably very subtle and that it may not affect the overall. Because as I say this, I don't think that there's much, this is all well before conscious processing. So there's certainly not going to be any conscious behavior input. And whether or not you could increase the synaptic efficacy through typical potentiation measures, I would guess that yes, but also that it's probably pretty subtle.
So one thing I was thinking about is maybe doing the startle response or something. So say for high visual acuity for objects moving close to you. I don't know if there's any tiny facet predators that prey on ferrets or anything. If you could via training or optogenetics, maybe either excite or inhibit this pathway to see
Right.
changes
What
in
would the
response
behavior
time.
implication be if you could selectively inhibit the pathway or not? Again, I think that this is a very subtle manipulation of the visual signal, just enough so that it probably helps prioritize visual signals that are relevant against visual signals that are irrelevant. So, kind of leading again back to Dave's question that if you could stimulate the pathway, could you get a major behavioral change? The answer, I think, is it would be very, very hard to see a behavioral change. I think it would be very hard to quantify because the nature of the feedback is so subtle. Yes, Barry.
So one of the popular ideas about the function of the system is that it has a role in attention. Do you believe that, or just, and that's a fairly subtle?
Right. So, yes, in fact, Vanessa's data that she showed yesterday, she thinks that maybe, that she shows some attentional effects between V1 and the LFP, particularly changes in coherence between the LFPs of V1 and the LGN. And it seems likely that the corticovernacular pathway is the conduit for those signal changes, because there's nothing else. So how I would go from this mechanism of increasing response, decreasing response latencies and precision, and how I would say that attention can kind of drive this, is that maybe the attention helps by specifically driving those corticovernacular neurons in the retinotopically aligned area, and so the process is top down, where for a given receptive field, for a given retinotopic area, I would guess that attention acts somewhat in the same way that our LED does, where it subtly increases the firing of those corticovernacular neurons, and then that leads to these subtle changes in the response properties. But I think that where we are is pretty reductionistic compared to the overall complexity of an attentional mechanism. That answers your question. Yes, Grandma.
Is there any effort to test, and I'll just use this very generically, the vision of the ferrets before you ever start? In other words, if you were working on humans, some of us start out with better vision than others. So
Yeah,
how,
that's a
do
good
you know
question.
whether you're measuring the same thing?
Sometimes I've sure wondered if my ferrets are blind. Every time that I've poked around for receptive fields and just for hours and hours I can't find them. Yes, there probably is a way to test for visual acuity in the ferrets. We don't really do that. We just kind of assume that their visual acuity is all about the same. One of the things that we do is we try to limit the distance our stimuli are from the ferret's eye, so that and that kind of takes away
Any variation.
that acuity issues that might exist. I imagine you could do some sort of a behavioral test where if you snap a clicker or something like that, you might be able to get the ferret to attend to it very quickly. We tried something similar to this in our lab, where for a project that sort of sank, we needed to get a picture of a ferret looking at something, because we wanted to get an idea of how the head shape, where the direction of the gaze is. For instance, if my fingers are the ferret's snout, does it kind of look in this direction, or does its gaze follow the aim of its snout? And so we tried very hard to have the ferret and try to get it to look at something and take a picture of it looking at something, and that was a dismal failure. But you could do something kind of like that, where I suppose you try some sort of a task. There are some ferret projects getting started where people are using awake ferrets to look at mechanisms, kind of like attention that Barry brought up, and these ferrets are being trained to do some kind of a behavioral task. I don't know much about how those are working, but I imagine if we can train mice to do it and rats to do it, then ferrets can probably do something like that as well.
So you showed the figure where you looked at the differences in response latency with and without LED, and you looked at the on neuron versus the off neuron, which you said was neurons that respond to increments versus decrements in light intensity. So it looked like you mentioned there was a slight difference in the response latency between those two, and I understand that was just one neuron that you showed, but
Right.
was that a pattern that you saw, and is there any physiologic significance to that?
So you're talking about the difference here,
Yeah.
this on neuron, the peak pixel frame is in the
Mhm.
20 millisecond frame, whereas this one is in the 40. No, this didn't reflect any specific pattern that we saw. However, this does show that there's some variability here. Part of the problem is the resolution of our end sequence. So each of these frames flashes at 20 milliseconds. That's the refresh rate. So when I say this is 0 to 20 milliseconds, this is the frame that was immediately preceding the spike. But because it takes 20 milliseconds for the frame to shift, it has to, there's some time there. We don't have resolution greater than 20 milliseconds. So in this case, what could have happened, especially since these two receptive fields are pretty close in brightness, is that this one maybe only took 25 milliseconds to respond, but we don't have the resolution to determine that, so it kind of ends up in this second window. Also here, if you look at this distribution of on and off cells, there's really not much difference. There's actually a little bit of a bias maybe in that some of our on cells had the greater reduction. So some of the on cells went from slowest or had the biggest change, but we didn't really quantify where that change started, whether they had the slowest start responses.
So this doesn't really translate to any physiologic difference with how we perceive, say, a light going on versus a light going off?
So our data does not really translate to that. There probably are some physiological differences that's based on the circuitry that generates the on versus the off responses. But I don't think our data really speaks to that. Steve, you had a question?
It was pretty similar. I was going to ask about the implications for ferret, but.
And Matt, you had a question?
Yeah, you talked about the stream-specific hypothesis.
Yeah.
Did you find any evidence to support the homogeneous piece, or did you find any evidence during the? I'm not sure if your experimental design supported the other one.
Well, you could say that if you remember on our pie chart, we had some neurons that had promiscuous connectivity, where they went from X-like corticogeniculate to Y-like corticogeniculate. That could be some evidence for some crossing information streams. We know a little bit about in certain primates that corticogeniculate axons sometimes target multiple different layers. So there's a third pathway involved in all this that I didn't really talk about because we didn't test it. In primates, that's called the coloniocellular pathway, and in ferrets, it's the W pathway. And there's been some evidence that corticogeniculate axons in primates that target the parvocellular layers will also send collaterals to the coloniocellular layers of the LGN. So there likely is a little bit of promiscuity here, but overall our data really does support a functionally stream-specific organization. All right. Thank you everybody for coming.
So pending successful defense, there's a party at 1 o'clock in the 7th